Hi, Dan.
Hi, Dan.
Uh, I think we're all here.
Incredible.
Yeah,
It's
I know.
57 and you're all here?
I know. This is what I was just saying. I was just praising all of them for being early.
Punctuality.
I know. They're so good. Um, all right. Well, we're going to get started. If you're ready, Dan, are you ready to go?
I hope so.
All right. Great. So what I thought I'd do is have the students introduce themselves first, and then you can introduce yourself and go on for as long as you like on background about yourself, and then we'll just open it up to questions. All right?
Sounds terrific.
Great. Simon, would you care to start?
Yeah. I'm Simon, and I'm a senior at the Head Roy School in Oakland,
All right.
California. Yeah.
Good.
Are you an Oakland A's fan?
No, not really. I'm not really a baseball guy.
I
All right.
am.
I don't know how. Zainab, am I saying your name right?
Hi, it's Zainab.
Zainab?
Yes, I'm Zainab. You can call me Zay.
Zay.
I go to the Dalton School in New York, and I am a sophomore.
Great. Abby, how about you next?
Hi, I'm Abby. I go to the Head Roy School with Simon in Oakland, and I'm also not an A's fan.
You guys Giants fans?
Yeah.
I grew up in Yuba City. You guys
Oh,
know when
nice.
it comes? Pardon me?
Oh, that was nice.
I guess I'll go.
Someone is typing, which is great, but if you are typing, it'd be also good to mute your microphone so that the typing sounds don't get picked up on the recording that I'm making. Go ahead, Tatiana.
Hi, I'm Tatiana. I go to Albuquerque Academy, and I'm a junior.
All right. It's over to you, Dr. Drollet.
So, how many of you are interested in medicine or biology?
I'm pretty interested. I've always found it pretty fascinating. I think really my interest in medicine just started from just as a kid watching House with my dad, and then from there I got more interested in it.
Anyone else? No?
Yeah, I'm definitely interested as well.
Yeah, so looking at some of the questions you guys sent me, it seemed like you were very curious about how I got interested in biology and how I ended up where I'm at. Sort of what was the path that led me to where I am now. And I'd like to tell you that it was all well planned and then, but I don't think that's really true for anybody. I was always fascinated by biology, and so what I brought was a curiosity, I think, and ambition to understand biology. And there I did the typical route that you guys are doing now, and I went to study biochemistry at UC Davis. And if you're interested in biology, the most important thing I did as an undergraduate was to work in a lab. And so I worked in a laboratory on campus during the summer between my junior and senior year, and all through my senior year. And I found that I really enjoyed working in the lab. And I think that the most important thing you can do is to test out your interest and see if you really like doing them day to day. When I graduated with a degree in biochemistry, I didn't really know what I wanted to do, whether I wanted to be an MD or go and stay into research. So I wanted to take a little time off. And so I started applying for jobs, mostly in the Davis area, working in a lab. And I wasn't very successful at getting jobs. It's hard. But finally, I got an interview at Genentech, which had just gone public. You don't know about this. Is way back in 1981. before everybody had a computer on their desk. And I got that position. It had just gone public. And this was the emerging biotech industry. And they were sort of like the biology equivalent of Bill Gates at Microsoft and Steve Jobs and Wozniak at Apple. It was this young, exciting company that was going to cure the world of cancer and other diseases through making human proteins to fight disease, what we call biologics. And it was a great place, pure luck. I was so glad I didn't get all the other jobs I interviewed with just to get this job. And it was fun. I remember the first week I worked from 7 a.m. to 12 p.m. every day because we were trying to make a milestone. We had to make a gram of this drug called interferon. It was a protein that was going to cure cancer. Of course, it didn't quite do that, but we all were hopeful at the time. And if we made a gram of it, we got $1 million. Back in 1981, that was real money. So,
Dr. Gillette, just a quick question. Just interrupting for just a moment. A gram, how much is a gram?
I haven't thought about ounces in a long time. You guys must know what a gram is.
I don't know. Some of my...
You know what a kilogram is?
Some of my students...
A kilogram is 2.2 pounds. So, so a gram is 1/1000th of a kilogram. So it's a very small amount.
And that was the goal, to make 1 gram.
To make 1 gram.
All right.
Of this protein.
All right.
And we achieved that goal. And so I was kind of hooked on this idea of developing pharmaceuticals. And because there was also a thrill involved when you actually did help someone in need, develop a new therapy. And I worked on all kinds of different drugs back then. I worked even on pharmaceuticals for cows. We worked on interferon for cows in order to prevent shipping fever in cows. This is where they would lose weight when you move them from the farm into the holding pens, and then the farmers would lose money because the cows would lose weight from disease. It's sort of like when you went to school every year, you'd get a cold in the first couple of weeks because all you guys have been away from each other for a long time, and then you get together and you bring the germs to one another and you get sick. That's what happens with cows when you put them in holding pens. So we also made interferon for cows, which was squirted up their nose. So we called it beef well again, and we called it cow cane, and because it was squirted up their nose. And we ended up selling that process to Monsanto. And so we did all kinds of things. Let's see, I worked on human growth hormone. I worked on proteins that helped make birth process easier. And so I was only going to work a couple of years and then go back to school, and I ended up being there for 6 years. It was so much fun. And then I went on to graduate school at UCSD, University of California at San Diego. And it was actually very hard to leave. It was so much fun, but I really had other goals I wanted to pursue. So I went into a doctoral program at UC San Diego. And I worked in a lab, I think I mentioned in the biography, at Jeff Rosenfeld's lab. We were studying transcription factors that help the pituitary gland develop. And so what's a transcription factor? Well, it's another protein. It's a protein that actually reads your DNA and turns it on at a specific place and a specific time. So I was looking for those proteins that would turn on a cell type called thyrotropes. These are the cells that make thyroid hormone in your body. Maybe you've heard of this. Thyroid hormone, many people are hypothyroid. They don't make it. Their metabolism slows down. So it's one of the master regulators of your metabolism. So when you don't have enough hypothyroid, you'll get cold and your metabolism will slow and you'll gain weight. All these bad things will happen. So we were trying to understand how the thyroid was turned on. And so that was my project. And so I was in competition with the group in Colorado where I'm at now, or so I thought. I once found out where my competition was with snuck into, that's one of the things that's exciting about this field is it is competitive and exciting, and you're really kind of racing the clock many times to finish up your work. So what can I say about that? That was so what it turned out is I thought I had found a factor that actually caused thyroid phenotype, or caused these cells to be born. It turned out later that wasn't true. It was actually, turns out that it's a protein a transcription factor that regulates your circadian rhythms. So that is, it controls your, well, you all know about circadian rhythms. This is why you can't wake up in the morning, right, at this age. Certain proteins are turned on in the morning, and other proteins are turned on at night. And so this is the 24-hour cycle that we all have. And this protein was involved in regulating that cycle. So that's some of the things that I sort of did in graduate school. And from there, I couldn't wait to get back into developing drugs because I had fallen in love with it at Genentech. And then I've worked at several pharmaceutical companies after that. Currently, I'm working on drugs for ocular disease indications, most notably something called age-related macular degeneration. Anybody hear of that before?
What is ocular disease?
Diseases of the eye. Has anyone heard of age-related macular degeneration?
My grandmother has it.
You're right. So it's the leading cause of blindness in Americans over the age of 60 years old. And until about 10 years ago, there was no treatment for it at all. And now we're just beginning to develop treatments to control it. So,
can I just ask a quick question, Dan, to interrupt?
Sure, interrupt all you want.
Okay, I just want just a question for Tatiana. Is this a coincidence that your grandmother has this, or did you select Dr. DroletSession because you saw this in his biography?
I just saw that he was a researcher, and then afterwards I read his biography, and I just saw that. It was funny.
Okay, so it is a bit of a coincidence then.
Yeah.
Yeah. All right. But as anything that's as common as this disease, it's as soon as you mention it, all of a sudden, you start finding it all around you. Go ahead, Dan. Sorry to interrupt there. You were telling us about AMD.
Yeah, it's the leading cause of blindness in people over 60, and it's not surprising that your grandmother, because the number one risk factor for getting this disease is growing older. That's the age part, but also the second biggest risk factor is actually being female. And that's not just because females live longer. If you even take out that factor, females are more prone to getting this disease, though it certainly happens in males as well. And so it's a disease in which the back of the eye, there's this layer of cells in the back of your eye that nourish the retina. And the retina is the layer of cells that actually absorb the light and process the light to signals to your brain. And there's this other layer of cells that care for that retina. And those cells start to die. And when they die, the retina above these cells dies. So it becomes a factor of why do these cells that we call the retinal pigmented epithelium, because we have to have a big name for everything, why it is that those cells die. And one of the reasons it's suspected is something called complement. And complement is about 30 proteins in your blood that fight off disease. And so the thinking is that these proteins become dysregulated with age. So instead of fighting off disease, they start attacking your own cells. And so my current research now is to look for factors that will stop the complement from attacking your own cells, or these retinal pigmented epithelial cells in the eye, and therefore to stop the disease progress. That's what I'm currently doing now. Any questions?
Why don't we look to some of the questions that you all prepared in advance? Does anyone want to offer one of those questions, or you could pick up on something that Dr. Drolet has said already? Simon, go ahead.
Well, just being a senior, looking at a bunch of schools, I'm just curious how you actually chose to go to the schools you chose to, and why you went to those for the major you chose.
Yeah, I was interested in biochemistry. I was one of the few students as a senior who actually knew and was passionate about what field I wanted to go in. And there were two schools at the time in California that were top schools in biochemistry. One at the time was Berkeley, and the other was UC Davis. And I chose UC Davis, which was a good choice, and probably because I didn't see myself as a Berkeley type at the time. But I chose it because it was a strong and well-recognized school for the major that I wanted. So it was as simple as that for me. I have a 16-year-old daughter, and she's beginning to think of schools, and she's interested in biochemistry, so we're going through the same sort of thing that maybe you are. And I think I think if you know what major you want to do, it's easier to do the search. If you don't, that's okay too, but maybe it's a little more difficult to find the schools.
Yeah. How about someone else, Abby, or? You guys, I have that document. You guys entered some questions. I don't know if do you want me to?
Oh, I know for a fact.
That's all right.
Go ahead.
No, I'm waiting for a question.
Well, I have a question, but I don't want to take up the students.
I'll go ahead and ask one.
Go ahead. Go ahead.
Well, my question is essentially just how long, like in terms of years, does it take to develop, like for example, interferon, or that drug you mentioned for the cows?
Yeah, was it you who asked the question about what's it take to get an approval from the Food and Drug?
Yeah, I was curious about like the process of just getting this out into like the market essentially, and like at what point is it okay to develop it, like and introduce it to other places?
Great question. It is it's a long process, and frankly, as a researcher in this field who has to get drugs approved by the FDA, I'm thankful that the FDA exists, because without someone making sure the drug is safe and effective, there'd be a lot of scam medicines. It'd be like all the internet things you see out there. I think you couldn't trust anything. That's my little political commentary for the day. But it takes a decade, sometimes more, from conception of an idea to the approval process. So if you start with a disease condition, say, for instance, one I know about very well, let's take macular degeneration, where a special type called the wet form of macular degeneration, this is where blood vessels grow from the back of the eye right into the retinal layer. They leak and hemorrhage, and they cause all kinds of damage to the retina. So you want to stop that growth of blood vessels. And there's a protein called vascular endothelial growth factor. So vascular meaning blood vessels, and this protein causes these blood vessels to grow. So if we can develop an inhibitor to stop those blood vessels from growing. So we did. A company I worked for took this on back in the mid-90s, 1994. Sorry, there's a train going by. So back in it was about 1994, we started that drug, and it was approved in 2004. So it was actually pretty quick. It was 10 years. So it took about 4 or 5 years to develop the drug, to identify something you want to put in the body. And so once you have such a compound, then you have to do a couple things before you can go into human trials. Before you can put it in a human, the FDA wants you to let them know that it is safe. That's the number one most important thing. That was a question I was asked. So the FDA cares more about the drug's safety actually than its efficacy. Its efficacy is how well it actually works or helps the person. It first do no harm. It's part of the Hippocratic Oath. And so that's what the FDA cares most about. So we have to do animal studies with our drug. It's one of the things that we do in order to make sure it doesn't harm the animals. And we also have to do efficacy studies. We have to show in animals that this drug has some ability to reverse the disease process. If you show those things, then you can file what's called an investigational new drug application, an IND, investigational new drug application with the FDA. And the FDA reviews that, and then it gives you permission to begin testing in humans. And the testing in humans occurs in 3 phases, called phase 1, phase 2, and phase 3. Now, phase 1 human clinical trials are all about safety. You put in a very low level of the drug into the human, and you ask if there's any harm being done. It's not about whether the drug works. It's simply about whether the drug causes harm. And so you have to get through that phase. It's a small number of people. Sometimes it's healthy volunteers. College students often volunteer for these studies to maybe earn some money, because often they would be paid to be participants in such studies. Sometimes it's patients who are in these studies. It depends on the type of drug and the safety of the drug and the disease indication. So once you get through phase 1 studies and you say, oh, the drug is safe, then you can go to phase 2 studies. And phase 2 studies really are about how much drug do you need to put in the human for it to work? And really, does it work? So those are called dose-finding studies or dose-ranging studies. And so here you have to show the agency. So in phase 1, you have to show the agency that the drug isn't hurting anybody. And then in phase 2, you have to show the agency, and by saying agency, I mean FDA, that the drug is actually doing some good. And still looking at safety. So now you come up with a dose that works. You have more safety information. And then you apply to the agency for phase 3 testing. And this is the type of testing most people hear about. Once you get to phase 3, you get these very large clinical trials to test which are blinded, of course. So what that means is you have a set of patients that don't receive the drug and a set of patients that do receive the drug. Now, we call it double-blinded studies because it's double-blinded because the doctor doesn't know which patient got the drug or the placebo, and the patient doesn't know if they got the drug or the placebo. So it's blinded to both. And so they do the studies, and then it's completely objective, or hopefully it's completely objective, about what are the side effects we see in the people with the placebo. And yes, there are side effects in people who get placebo. And what side effects do you see in people getting the drug? And we also see, well, what are the responses in people getting placebo? And yes, there are responses in people who get placebo. And what are the responses that people getting the drug? And then you compare these things through an analysis to see if actually in this large patient study that they're still not done, because the FDA requires you to do 2 of these studies, not just 1. You have to do 2. It has to show that it works and is safe in 2 completely separate studies, unless there are exceptions. If it's clearly an unmet medical need in which people will die without the drug, then sometimes the FDA will allow you to start giving the drug to the general population before you complete the second study. But they still require you to do that second study. So all of that takes about 10 years to accomplish, sometimes a little faster, sometimes a little slower. But to get through all of it, maybe only about 1 out of every 10 drugs that go into human testing actually is approved by the FDA. So there's a lot of failure in this business, and you've got to be somehow come to grips with that you can maybe work for a long time on something that doesn't work. But that's also why you have such a thrill when you work on something that actually does help people in the end.
Actually, Dan, that relates to a question I wanted to ask. Most of the other speakers and most of the topics of this course revolve around, one physician treating one patient at a time, and in their career, they may treat many hundreds, maybe a few thousand patients. I wonder if you could contrast that with the kind of satisfaction you were just referring to. Do you know what I'm asking? What I'm saying is, you were saying that when it works, it can be very satisfying when you actually find something that is effective and safe, and people are being prescribed a new drug. What I'm asking is, I wonder if you can contrast the benefit that a great doctor does in his or her career for the hundreds or maybe thousands of patients that they see, with the benefit that someone who develops a new drug might do in his or her career. You see what I'm asking?
Yeah, I think so. Let me try and answer, then you can tell me if I was successful or not. Mostly we're anonymous back here. So I think mostly the pharmaceutical industry gets, why is your pill so expensive? It's so cheap to make. That no one really thinks about how much research and how much research time and the people who brought such a pharmaceutical to the marketplace. And so our rewards are a little more impersonal. We take pride in what we do, but unlike a physician who actually is seeing the patient, and these people are wonderful. I have nothing but praise for physicians who practice the human more of a human art than we get to practice often. We're doing a lot of our stuff in the labs. We work with doctors. Actually, it's even in the clinical trials I mentioned, it's the doctors and nurses who are giving those drugs, not us. We get it in the bottle or we get it in the pill, and we give it to these centers where the clinical trials are going on. And the doctor who's trained in medicine then administers these drugs. So they have the reward of the personal side, seeing the drug directly help the patients. We have to accept ours a little more on the impersonal side. But we know that what we do can have a tremendous benefit, and that keeps us going.
I guess what I was kind of hinting at is that...
I missed it, huh?
Oh, no. That you've answered my question. No, you've totally answered my question. But I was also trying to hint at just how much any one doctor's success would be dwarfed entirely. Even the most successful doctor who has ever practiced medicine doesn't do a tiny fraction of the good in the world that is done by someone who invents a really terrific drug. Right? If you invent a oh, come on. If you invent a life-saving drug that tens of thousands or maybe millions of people are receiving, a new antibiotic, a new anti-infective, how can you not compare yourself well against any physician in that arena?
I hope my answer didn't say I wasn't comparing myself well. I was contrasting the rewards of it. But the rewards are different, and it's a personality sort of thing. I did think about medical school, but I loved the researching more, so it was a personal choice for me. I really enjoyed working in the lab. I really enjoyed this aspect, and so the enjoyment of doing the work is also a reward. And if your goal is to be a physician, and that's gives you pleasure and the reward, that's important. I don't know how to contrast how much benefit one person does over another. I like to think that we've done more good than harm.
I have a question.
Sure.
So, both of the disease that you mentioned, the ocular disease and pituitary disease, both of those are pretty common. And I was wondering how you balance in your field the financial rewards from finding a drug for a really common disease that would affect lots of people, or finding a drug for a much more rare genetic disease that maybe people are dying from that could be helped.
That's another great question. And frankly, in our industry, we don't do that very well. Obviously, the way our system works is that we're a capitalist society, and companies are set up to make money, and and so they often will spend their research dollars on the biggest problems. It's not necessarily a bad thing. They are, after all, the biggest problems.
Yeah.
So, for instance, someone asked me it's a question about where the research dollars are spent. Even our government spends most of its money on the big problems. The number 1 and 2 researched area are heart disease and diabetes. Why? Well, most people die of heart disease still. Almost 50% will die of heart disease. It dwarfs cancer. Diabetes is 1 out of every 5 healthcare dollars now are spent in diabetes. So treatment, medical costs, 1 out of every 20% of all healthcare dollars. So those are the big ones, and those are where the money is mostly spent, and those are where the drug companies go after. Now, in cancer, there's 4 big ones: breast, prostate, colon, and lung cancer. So most of the research goes into those cancers. But that's not to say that the others are completely ignored. There are a company, one called, dropped out of my head. I can't think of the name of the company. That's embarrassing. Starts with a Z. Anywhere that does work on the rare diseases. It's a business model that they go after all the rare diseases. So how do we do that? Actually, the government gives us incentives to spend more of our research dollars on the rare diseases with what's called the Orphan Drug Act. It's a funny name, but what it does is it gives incentives, monetary incentives, to drug companies in order to go after the more rare diseases. The other place that you've probably heard of where some of these rare diseases are being funded is through the private donations, the Gates Foundation, and other foundations that give money to underfunded research into disease. One of the biggest problems we face are third world diseases, which there's no money in at all. So Gates Foundation and others are actually the ones who are pushing the study of malaria and tuberculosis and even Ebola. You've seen there's been a lot of donations into Ebola. It's really, not a threat to the U.S. healthcare system as yet. There's been some cases, as you've all read in the news, but should we throw all kinds of money at Ebola? Well, that's kind of hard since it's not a threat much in Europe or the United States, and certainly Africa can't pay for it. So that's where these foundations have stepped up to the plate and actually funded research. It's not a perfect system we live in, but this is how it gets done today.
Great question, Abby.
Question.
Yeah.
I have a question.
Yeah.
So in your biography, you said you're trying to use modified DNA for macular degeneration. So do you map out the DNA, like figure out where the mutation is, and then try to fix the mutation? Or
That's an area of research, but that's not quite what we do. We actually use DNA as the drug itself, but we don't go into the cell, so we don't modify the genetic code. We use these things as inhibitors. So in other words, this should blow your mind. So let's discuss possibly evolution for a second. So what came first, the DNA or the protein in evolution? What's thought was that there was the RNA world. Are you familiar with the central dogma hypothesis that DNA codes for RNA, which then makes proteins? Are you guys all familiar with that? Is that a yes?
Yeah.
Well, so if you ask yourself, well, what came first? How do you get that? Well, there's something called the RNA world hypothesis, RNA. So what that is that the RNA actually began by serving both as the genetic material and as protein substances. In other words, the RNA itself could catalyze reactions in the body for metabolism. The RNA itself could then bind to, actually link to another RNA or a metabolite and actually direct its function or use in the body. So RNA actually can bind to proteins. Everybody thinks it's proteins binding to RNA. So do you guys know about the ribozyme, the protein making factory in the cell? Have you read about that?
Yes.
Yes.
So that's an RNA complex, and it binds to proteins. So right there, we may have the remnants of this dual function of a nucleic acid, that it not only can code and be the genetic code, it can actually be a player in the game. It can actually be running metabolism, doing these sorts of things. So we take advantage of that property, and we then could take bits of RNA. These are about 40 nucleotides long, and we find ones that actually can bind to a therapeutic target, like a protein that's expressed too highly in a disease or something like that, and actually block its function. So, but we don't use natural DNA. We use what we call modified DNA. We actually,
Dan, let me just can I just
am I too technical?
No, I think that was terrific. I think it'd be helpful if I just show with my hands. So, if my left hand here is a protein involved in some disease, and my right hand could actually be a form of DNA that, rather than being shaped like a double helix, that, kind of boring shape, has some complicated shape. Dr. Gillette's research involves taking a complicated, complexly folded DNA sequence which binds to the protein and therefore prevents prevents a disease, or I think Tatiana's question was also about identifies that protein as in the biomarker application of this technology. So is that all right, Dan? Did I say?
That's good. Yes.
Okay. And actually, Dan, can I ask you just since I've interrupted, if you could just maybe scoot your chair up a little bit.
Oh, closer.
There's a distracting light coming in. It's on your shoulder now. It was on your face earlier.
Oh.
That's better. Thank you.
Well, some people say that's a good thing.
No, no, it's like a line of light that was.
Yeah,
it was.
the sun, I've got all the blinds closed. The sun is directly in my eyes right now.
That's perfect. If you're comfortable where you are now, this is perfect. Sure. So sure. Tatiana, I don't know so let me turn it back over to you. Tatiana, could you actually ask your question one more time, just so it's clear?
Okay, so I asked if they map out the DNA, figure out where the mutation is, and then try to fix the mutation.
Yeah, and as I said, we're using it more, as Dan said, in which we actually use them as missiles, if you will, to destroy a specific target within the body, that is not the genetic material, whether it is a protein or some evil metabolite that's causing damage, some chemical in the body that we don't want there, and we use it to intercept that and block its function. It's wild in the sense that we're showing that nucleic acids are more than just the keepers of the code. They actually are playing the game, if you will. They're actually can direct the metabolism itself directly and not through protein. I hope that's clear. And we're using it that way. And that's the basis of the technology that SomaLogic has developed. So now we're using this not only as a potential for therapeutic, but we're using it for biomarkers. So now this is the two facets of these bits of DNA. They can not only bind to a target, actually tether themselves to a target. So imagine that I have 1,000 or 2,000 of these bits of DNA that each bind to a different target. So we can then take a bit of blood, put it in a test tube, and then add these 1,000 or 2,000 bits of DNA, and they bind to the target in there, each a different target in your blood. Then what we do is we separate those things that are bound together, tethered together, from the things that aren't tethered together. gets in your blood, we can quantify at the same time. And with that, we're trying to use this technology to understand when someone might have a disease or whether a treatment is affecting the disease in the way we want. So we think our technology is different in that genomics, or what you've all heard about looking at the genome, that might tell you, oh, this person's going to get diabetes. Maybe someday they can say that. Or this person is prone to breast cancer. This person over here might get colon cancer. But obviously, these people lived several decades without such a disease before whatever that genetic thing is that they have may or may not trigger the disease. And so why is that? When is that? The DNA can't tell us when it'll happen. We're hoping that the proteins in the body then are the signature. They're the ones that can tell us when someone who's prone to maybe diabetes because they have this mutant gene will actually become diabetic. And with that, that's what we're using our protein for. And someone had asked, when will that be in the home, that you can actually do this yourself? And actually, that's the dream of one of our chairman of the board he calls it the wellness chip. So he has this dream that everybody can take home this chip of these DNAs and actually can measure at home whether they are still well this year, do it once a year. But that's maybe decades into the future. But right now we are having success with it in a more limited scope than you doing it and saying, oh, you're completely healthy. But before I get into that, let me just say that someone said, oh, when will these protein analyses be available, like 23andMe, the genetic kit? Well, actually, protein analyses have been available. Everybody knows of the pregnancy test strips that you buy at the store and you pee on, and it tells you whether you're pregnant or not. Well, what that test is, it's measuring a protein called human chorionic gonadotropin. It's a mouthful, so we say HCG, because it's too hard to say that 3 times fast. So when you do that test, it says, in your urine, is there that protein? And that protein is an exquisite signature that you are pregnant. Now, let DNA tell you that, right? DNA can't tell you by looking at your genome whether you're pregnant or not. But this protein will tell you instantly whether you're pregnant or not. So that's the kind of thing that we're looking at. Recently, one of the breakthroughs that we've had with our technology, where we measured 1,129 proteins simultaneously, was a study in mice using parabiosis, it's called.
Oh, yeah.
This is really wild. This goes back. What?
No, I'm just excited to hear this story again. I never tire of this. Go ahead.
This goes back 160 years that people have been taking mice and stitching their blood supplies together. You could actually take 2 mice, put them side by side, and connect their blood vessels together so that they share one circulatory system. 2 hearts, 1 circulatory system. And people have been doing experiments like that for a century and a half. And one of the things that had been noticed was if you take an old mouse and you do parabiosis or mix the blood supplies with a young mouse, then actually the heart of the old mouse will get better. It'll more resemble a young mouse. And the question is, why is that? And for over 100 years, we've been wondering why that would be. What is it in the blood of a young mouse, if it is something in the blood, that makes an old mouse's heart better? And of course, we know that 50% of all deaths still, or roughly 50%, are due to heart disease. So this is one of the big problems. And this group at Harvard, by Amy Wagers and Lee and professor, I can't remember his name right now. They were studying this and trying to identify what this factor is, and meeting no success. And so they asked us to look at the blood of old mice and young mice and find the differences. And we found several differences, including this one protein called GDF11, which is growth differentiation factor number 11. And it was much higher in the young mouse than the old mouse. And what they were looking at was the heart. So let me explain a little bit about the old heart. When the heart gets older, it has something called hypertrophy, a fancy word again, which means the cells in the heart are getting bigger. They're getting abnormally large. And what that also causes is a thickening and a slowing of the heart. And the heart actually gets heavier than it should be.
Actually, we just finished a case on cardiology, and our patient did have an enlarged heart. So they're somewhat familiar with this. Go ahead.
Great. So what they did was say, okay, well, here's GDF11. What happens now if we actually take the protein, not do parabiosis? So the hypothesis became, I put these two mice together. The heart got younger. One of the old mouse, the heart looked more like the heart of the old mouse, looked more like the heart of the young mouse. Could it be the GDF11? So this is what we all do every day in the lab. Well, that's the hypothesis. So now how do you test whether it is GDF11? Well, you take an old mouse and you put in the protein, not just the circulatory system. You leave the young mouse out of it, but you just put the protein. And lo and behold, when they put the GDF11 protein in the old mouse, its heart got lighter, and the hypertrophy started to disappear, and the thickening got less. So it works in mice. Now the question is, and what other species besides mice? And if it works in another species besides mice, anybody there?
Sorry, Dan. Yeah, things you got a little bit broken up there. I'm here. Can you hear me?
I can hear you now.
Okay. And is everyone else frozen? You're frozen on my screen.
Okay. Well, I'm not touching it.
Yeah. No. You don't need to do anything. This is the sort of thing that just happens, and
Simon's gone too.
Yeah, same. Simon's gone for me too.
Simon. We've dropped Simon.
We can wait.
Yeah, I'm going to try to bring Simon back in. Give me a second. Simon, are you there?
Yeah. Oh, yeah.
Sorry. Simon's back. Okay, it looks like
Hi, Simon.
we're back. Okay, we just finished injecting GDF11 into an old mouse, and it made its heart young again.
Ed, are you gone again?
No, we're here. Everyone's here.
Okay.
We're all here.
You're breaking up. All right. So I was saying that the next thing people will do is maybe look at a different species besides the mouse in order to determine if this is a mouse-specific phenomenon or if it works in another species, and then begin the drug development process to see if GDF11 might eventually be a treatment for heart disease. And so this will be a race now to see if we can get this thing approved. Not us. Somologic is not working on this, but other companies will be, and they'll take it through the approval process to see if this will actually be helpful in humans. And we hope it will be. It's very exciting to actually find a single protein that can reverse the decline in an aging heart.
All right. Terrific. Well, I think we should maybe ask for any one or two short questions, if we have any, and
I actually have another question.
Okay.
So this is sort of based off of, you mentioned earlier about how difficult it is sometimes to get funding for diseases that aren't as common. And that sort of reminded me of just over the summer, just in light of this ALS Ice Bucket Challenge,
right.
I was wondering what your opinion on that is, first of all, and also how effective it really was in terms of fundraising.
Oh, I don't know how much funds that they raised, but I think all of these things are wonderful. I mean, it's a way for people to become involved in the research themselves, in a way, supporting the research, and certainly ALS has been a very difficult disease to come up with any sort of cure. So you picked one I'm not particularly familiar with, so I don't know how to answer that directly, but
I think
I'm happy about it.
I think that the question, tell me if I have this right, is the nature of the question is when the public does get involved, they may, without a lot of expertise, cause resources to be devoted to diseases that might not be the highest priority. Is that part of what you're asking?
Not exactly. I'm
Okay.
not sure how high, how much of a priority ALS is. I'm assuming that it's it's not really about how important it is in terms of how many other diseases are out there. I was just wondering how effective movements like that are.
I see.
For diseases.
Yeah.
That's a great question. There's this argument that directed research is never as good as serendipity, the ability just to follow your nose and actually find something. The Fleming and the moldy bread, maybe you don't know this story about how this researcher named Fleming threw out this moldy petri dish in which the bacteria didn't grow near the mold. And then he realized that, hey, why didn't the bacteria grow near the mold? And that was the discovery of penicillin. He wasn't trying to find penicillin. He was just being a good scientist and a good observer, even more than a good scientist, and saying, hey, how come nothing grew near the mold? And maybe the mold is producing something that kills bacteria, and can we use this? And that's sort of the serendipity of science, that you find a cure just by doing good research. There have been successful directed research in the past. Probably one of the most famous examples is polio, with Jonas Salk and others, and there's the Salk Institute now in La Jolla near San Diego, a research institute named after this man who there was directed research in order to come up with a vaccine against polio. And part of that also was the March of Dimes, which was one of the very first one of these public fundraisers, which was supported by Franklin Roosevelt.
Right.
So people would give dimes across the nation to focus research into this, and that was successful. And so it can be. It also can cause money to be spent on perhaps less societal problems, maybe rare diseases. But sometimes if that's the only way to get any resource on that disease, then that's the mechanism we currently have. So it can be a two-edged sword, is what I'm trying to say. A little history lesson, maybe. Huh?
Yeah, no, that's actually really, I hadn't really made that connection until just now, that the ALS Ice Bucket Challenge is really nothing new, that the March of Dimes is a great historical precedent for what happened with ALS.
Well, there's the Conan Foundation, the Race for the Cure that everybody has, which is a emphasis on breast cancer. Though I have to say there's never been a limited funding for breast cancer, in my opinion. It is one of the big four. People spend a lot of research dollars on that. even without the Conan Foundation. But it's certainly one of the possible advantages of these foundations is they could use different criteria than the federal government or drug companies to pick research. So drug companies will pick what research they want to do for a variety of reasons. And let's be honest, some of them are financial. The NIH, or the National Institute of Health, will give grants to researchers in universities based on another set of criteria. And so when there's a foundation and they're giving money to researchers, they can choose a different criteria in order to award funds. So in that way, it could be good.
All right. I think that's all the time we have. I hope we can all just sort of say a group thank you to Dr. Drolet.
Thank you so much.
Yeah, I hope that was helpful.
Thank you.
I really enjoyed it.
Great. Thanks. So did we. And I guess I'll have the students log out now. And Dan, if you wouldn't mind, I'll just keep you on the line for just another minute.
Okay.
All right.
All right, thank you.
Thanks.
All right, good luck to you all.
Oh, Dan, thanks so much. That was great.
I hope it was good.
Yeah. Yeah, I could really see that the GDF-11 story was really interesting. I could see there was some interest there, and anyway, that was terrific. I thank you so much. I really enjoyed it.
Good.
Good. All right. The only reason I'm keeping you on is to just remind you of tomorrow's 3:30 call. And just check if that's, we're all good with that?
We are.
All right, terrific.
We are, we being me.
All right, great. Thanks again, Dan.
Yeah, it was fun.
You're so generous to come in on a Sunday for this.
Well, it was only because it was 2 degrees when I was shoveling again. That's what's hard.
Oh my gosh, you had to shovel your way out to get here?
No, I shoveled in the morning, of course, when it was 2. It stopped snowing and it's clear. You know how it's beautiful here?
Yeah.
When, after it snows and the sun finally comes out. But it's only going to be a high of 20-something today.